My name's Edward Hookway. I'm from the Nuffield Department of Orthopaedics, Rheumatology and Musculoskeletal Sciences at Oxford. We call it NDORMS for short. You'd originally invited my supervisor, Professor Nick Athanasiou, who unfortunately has had a long-standing commitment today, and so I'm afraid you've got me instead. But hopefully we'll be able to tell you some of the exciting things that we're doing with the support that we're getting from you. We're based here at the Botany Research Centre in Oxford, which is on the same site as the Nuffield Orthopaedic Hospital, where some of our sort of bone cancer treatment gets done, and we're basically in the car park, but it gives us a great interaction between the basic scientists, which is what I am now, and the clinicians who are dealing with people day to day. This chap here is Beethoven, and it was a little bit of his work that you were just listening to as I walked up. The music is from one of his string quartets, string in A minor, opus 132, if anyone's interested. It's a fascinating story. So one of the things that everyone knows about Beethoven is that towards the end of his life he started to go deaf. But as this deafness was progressing, he had one moment where he got his hearing back. And it was his experience of this that inspired him to write the music that you were just listening to. And I think it's just a reflection that, you know, illness can affect us all in a very individual way, and we might not all be able to express it in a way like Beethoven does, but it's a very individual experience that those of us who are working at the very basic science end of things need to remember that actually it's affecting people out there, everything that we do. I was asked to give a talk and given the brief of not to be too sciencey. Now, I'm a scientist, so I've tried my best. If anything doesn't make any sense, please shout out and let me know. We're going to start really from the basics, talk about DNA and cancer, a bit about how DNA is organised, and then go on to this thing, epigenetics. Can I just ask, has anyone heard of epigenetics before? A couple of hands. I'm really excited about this, and I hope by the end of this talk you'll all be excited too. So these chaps here are Watson and Crick, and they're sort of stood there very nonchalantly posing with their structure of DNA, which they solved 60 years ago to this year. And DNA is something that we all have to be interested in. The human genome is 3 billion base pairs long. That's a lot. And it's just made up of 4 very simple things, A's, T's, C's and G's. Each cell that you have has 2 copies of your genome. You have about 10 trillion cells in your body. And if you were to take all of your DNA and stretch it out end to end, you can get to the sun and back 67 times. That's a lot of DNA. And this is the thing that is sort of fundamental in the blueprint to everything that we do and how our body works. After DNA, when we're trying to have our body and how it works, we have something called messenger RNA. This is able to read DNA, and it forms a message. And this message goes from the nucleus of the cell, where all the DNA is stored, drifts out into the cytoplasm, the bit around the nucleus, and it can also undergo some various editing things. And this is a code. So each bit of DNA forms some RNA, which is a code, and every 3 letters of your mRNA codes for one of the amino acids. Now, the amino acids are your building blocks of protein. And proteins are basically what do everything in your body. So they're involved in many different things. So they can be structural, they help hold everything together. They're involved with enzymes. These are the proteins in your body which catalyze reactions to make things happen. They can act as a storage molecule for storing energy and other structural molecules ready to be used. And they're also used as hormones. They can send signals from one side of your body to the other. So we have a model here, which is DNA makes RNA makes protein. And that's sort of the fundamental concept which I will be going on about quite a bit today, I'm afraid. So DNA and cancer, what goes wrong with our DNA which leads to cancer? We've got a schema here which is talking about mutation. So by mutation I mean any change in your DNA which is different from normal. And because of the way the codes work, a change in your DNA leads to a change in your RNA, which can lead to a change in the protein. And as far as cancer goes, this can essentially have 3 different effects. The one in the middle there is it could do nothing. Some of these changes are relatively harmless, your body doesn't notice it, it doesn't make any difference. But it's the ones at the top and the bottom that we worry about. So sometimes a mutation in your DNA, a change, will cause a change which causes a protein to gain a function and start to drive cancer. And this can be in many different things. It could be in something that promotes cells to divide, and it's something we need to be really careful of. The other changes that could happen is you could have a change of something which loses function. Your body is amazing. It's got loads of different mechanisms for trying to prevent cancer growing within you. And sometimes these changes that happen stop one of these from working. Let's try and give an example. On the left here, we have the code for just one gene. That's about 2,500 letters long there, but that is still less than 1/1,000,000th of your entire genetic code. I hope you can see at the bottom there, we have one letter that is standing out, which is a big red T. This is a single letter that changes in one of these genes, a gene called BRAF, which is involved with many cancers, and especially cancers of the skin and melanoma. And we have a single change in this DNA, from that T there to an A. DNA makes RNA. We have a change in the RNA. We've got a table here which basically shows unraveling the code. And we change from something in the bottom left corner, what's called a valine molecule, to something further along to the right, a glutamine. And this causes a change in the protein. It makes the protein more active, and this can drive the development of cancer. So this is one letter out of your 3 billion that's changed, and this can start to drive your cancer. So dealing with your DNA is something that is fundamentally important to understanding how cancer goes and cancer works. So I've mentioned that all of the DNA in your body could stretch to the sun and back 67 times. How does it fit inside us? We have little proteins called histones, and these act like scaffolding for DNA. So our DNA starts off unwound, and it starts to wind around these proteins, which are represented here by the pink balls. And as they wind around, these proteins can condense into each other, and they start to form a much denser structure, which helps with your organisation. And these structures all condense down together, and eventually form what are called your chromosomes. Now this is a normal human complement of chromosomes. We have 22 pairs of what are called autosomal chromosomes, and then you have what are called the sex chromosomes, your X and your Y, and broadly that determines if you're male or female. So here we have an example with 2 X's, so this will be from a female. Chromosomes themselves can be important, especially in primary bone cancer. One of the driving factors in Ewing's sarcoma involves changes in these chromosomes. So these 2 of the chromosomes manage to swap part of their DNA with each other. As we've said, a change in the DNA leads to a change in the RNA and leads to a change in your protein expression. And it forms the new protein called the EWS fly protein, most commonly, although it can form other proteins as well. And it's this change which drives Ewing's sarcoma. So now we get to epigenetics. What is epigenetics, and how does it differ from everything I've just told you? A bit of Greek, epi, above, and genetics, the genome. And basically epigenetics refers to a way that the cells within your body control which bit of your genome in any individual cell is read. So we have a copy of everything in your DNA in each individual cell, but what makes one cell want to be muscle? What makes one cell want to be a bone? What makes one cell want to be your heart? These are controls which happen, amongst other things, in what we call an epigenetic fashion. And we have a definition here: an epigenetic trait is a stable, heritable phenotype, which means it stays the same and carries on, resulting from a change in chromosome, so that's the chromosomes that we've shown you there, without a change in the DNA sequence. The histones, which originally I showed you as little pink balls, are actually a lot more complicated. You can see the arms coming out of the sides here, and each of those little dots represents an area of the histone which can be changed. And this causes a change in the way that your DNA can wrap around it. And some of these changes make the chromatin, the chromosome, condense down, as we can see at the bottom. And there, it's impossible for your body to read those genes because they're all packed away tightly. So effectively, these genes don't get expressed. However, other changes open everything up and allows the expression to be increased. Now, one of the things that we know to start with is that the processes which regulate this in cancer cells has gone awry. We don't fully understand why it has, but we know it does. So one of the things we want to know is, by targeting these mechanisms and reverting them back to what they should be, can we find a way to help try and treat cancer? So whereas our genetic model had a change in DNA leading to a change in RNA to a change in protein, an epigenetic model we can think of more as we have a normal gene which is either silenced, perfectly normal genes, nothing wrong with them, can be overexpressed, there's too much of them there, and this themselves can help drive the cancer process. This is a cake. I'm going to try and explain everything I've just said in terms of cake. If we have a genetic cake, where we have our change in DNA, change in RNA, change in protein, I want you to think of it as if our cake had a change in the recipe. The recipe is the DNA. This leads, if somebody has written it down wrong, if there's a change, it leads to a change in the ingredients you put in there. You might double the amount of sugar, half the amount of eggs, do something crazy with it. And this will lead fundamentally to a change in the cake that you've baked. Now, it could be perfectly fine. It could, however, taste awful. That's like a gene which is not functioning properly. Or it could fail to rise and just sit there in a big gloopy mess at the bottom. This is a genetic cake. An epigenetic cake, however, is we've got a perfectly good cake recipe. But say you put that away in your recipe book, locked it away in a drawer, and thrown away the key. You now can't find the recipe for that cake, so it doesn't get baked, and you've got no cake. That's not good. Alternatively, as happened with I think it was one of Nigella's Christmas cakes a few years ago, everyone starts cooking it, and you end up with too much cake, and that as well is not good. I'm sure you've all read Matilda by Roald Dahl. Remember Barry Bogtrotter caught eating Miss Trunchbull's cake and she force-fed him the whole lot of it? Not good. So underexpression, overexpression, all of these are bad things. My work focuses on just two of these very small changes, which I've circled on the red here. They work in slightly different ways, the details of which aren't really important for today, but basically changes in these cause a change in the level of genes which are expressed within cells, and we know that this change happens in cancer cells in a way that causes damage. This is what I spend my life working on. It's a small molecule here, it's called GSKJ4. The GSK is from GlaxoSmithKline. We work in collaboration with a number of drug companies, and this is one of the molecules which they initially developed and then have put out for access for the research community to try and work on and understand. And this chemical works in a complicated way, but the long and the short of it is it alters the way that the chromosomes are packed, which is what we're interested in. Now onto some data. Scientists love data. I've tried to keep it as short as possible, but this is important. We have here some Ewing sarcoma cells. On day 1, we put the same number of cells into the left-hand side and the right-hand side. On the left-hand side, we gave them our drug. On the right-hand side, we didn't. And 48 hours later, hopefully you can see that there's a big difference. The left-hand side, the cells haven't grown. They're sat there, not dividing. That's good. On the right-hand side, they've done what unfortunately cancer does and spread out in a horrible weed-like pattern and have grown. So we can see, just looking down our microscope, that this compound stops the cancer from growing. Scientists are funny thoughts, and when we see something once, we don't believe it. So we do it multiple times in multiple different types of cells. Again, the details of these graphs aren't important. These are 8 different types of Ewing sarcoma cells, though, and it stops all of them, which is good. We then got really interested and spread out looking at the different kinds of cancer that this might work on. And we found that multiple myeloma, which is another cancer, not a primary bone cancer, but it affects your bones, stops that too. And colorectal cancer as well seems to stop that growing. But interestingly, it doesn't stop all the cancers. So we know we're not just giving something horribly toxic which kills everything. We're giving something which is selective. And it doesn't seem to affect normal tissue, doesn't seem to affect all different cancers, but it seems to affect some cancers in a really significant way. This is a pretty picture which I've included more because it took me hours to make. And I wanted to have somewhere I could show it to you. Again, the details of this, we're looking at 15 different types of cancer here. And the colours, the blue colours there, show the level of change in the expression of certain genes in response to our drug. The thing that's really exciting here is we're causing huge changes in the expression. In some of our cells, within 6 hours of giving this drug, we see the level of certain proteins go up over 1,000 times. And this is associated with the cells dying. You know, we're really excited about this. And what's equally exciting is it seems to be acting in some regards in our Ewing cells and our myeloma cells in very similar ways. So it's helping us to really dissect down what's going on here. Here's another pretty colour thing, which didn't take me quite so long to make. The important thing to note here is on the left-hand side we have some Ewing sarcoma, the first 2 columns. On the right we have our myeloma. And you can see that the blotches of red and blue, red meaning something is going up, blue meaning something is coming down, don't quite match up. And this is in keeping with what we see. We see our cells actually respond ever so slightly differently to this drug. The Ewing sarcoma cells just tend to sit there. They don't divide, they don't appear to die, but they don't grow. Our myeloma cells, however, we push actually into we reignite their mechanisms which cause the cells themselves to realise there's a problem and causes them to shut themselves down. So we've got some slight differences, but both are causing the cancer not to grow. So this is very early work. I've said, you know, it's a promising new frontier in treatment. Hopefully in 10 years it will be. You know, this is the very early work that we're doing in a dish. I'd love to say that we can get a treatment out there to help people soon. It's going to take a long time. We've still got lots of work to do. It helps us to understand what's wrong with cancer though, and by understanding what's fundamentally gone wrong, we can help to find new ways of trying to target it specifically and cure it. The picture on our right here has shown what I've been talking about. It's our DNA, the white cell in the middle there, which is dividing. This is a cancer cell dividing, which we can stop with our treatment here. The future work, we need to understand exactly what happens. So far we've only worked in dishes. We need to get samples of real cancer, which is why engaging with the sort of the research side of things when you're being treated is so important to us. We know probably it's the last thing you're thinking of if you're going in and you're having treatment, but if somebody has a chance to come and talk to you about research, it really helps us. And by helping us, you're helping everyone else in the future. And we are so grateful for all of the times we can get primary samples, so cancers from individual people, to do our experiments on. It really does make a difference. We're also going to actually move on and try and do this sort of work in some animals as well, because that's the next stage we need to go into developing treatment, and hopefully that's going to be starting very soon. The picture I put up here is the rather depressing space I do all my work in. So I need to be careful with what I say here because we're in a room with no windows, but it is a small, pokey room with no windows. That's an extractor fan, so if you're doing any work, you can't hear anything going on. You can't tell what time of day or night it is, and the air conditioning doesn't work. This is the lengths we go to because we get excited about this. And I'm sure none of you thought the life of a scientist was glamorous, but this is the reality. It's not all coming and glad-handing at conferences like this. Many, many people are involved in what we do. This is just a small number of the people who have been directly involved with the work I very briefly mentioned. We have various people in Oxford. We have people working in Birmingham, in Strasbourg, all over the place, and we get support from a number of people, the Structural Genomics Consortium, who help work and make these targets that we use. Santaris, one of the drug companies, are helping us with some work which is related to this. National Institute for Health Research, many, many others. But most importantly, it's you guys who are really helping us, and I'm excited about this. I really hope I tried to make you excited. I'm happy for any questions if we have time.